I hope you guys can see the slides okay in Amarillo and at the other stations. My talk today, some of you probably, I'm sure, caught the title, but I don't know, some of you didn't. You go to the field, U.S. Borlaug Fellows in Global Food Security. So Norman Borlaug, when he was asked, how do you become a successful wheat breeder, he replied, well, you go to the field. You go to the field again, and then you go to the field. And when the wheat plants start to talk to you, you know you've made it. Norman Borlaug, among the many accomplishments in his life, was awarded the Nobel Peace Prize, the Congressional Medal of Honor, and the Presidential Medal of Freedom. People have referred to him or called him the father of Green Revolution, agriculture's greatest spokesperson, and the man who saved 1 billion lives. So in the honor of Borlaug, the Purdue Center for Global Food Security has applied for a grant, and what it does is, through the U.S. Borlaug Fellows Program, it allows students to go to the field in their own right and to see an attempt to try and make a difference within their agricultural communities. So the U.S. Borlaug Fellows Program, like I said, is based out of Purdue. It's funded by USAID to expand the pool of U.S. food security professionals who have scientific-based needs to effectively study and manage global landscapes in support of sustainable food systems. And you know, it seems like a really big stretch, especially when you're a guy in Texas and you're trying to go to a foreign country to do some work, and it doesn't seem like it hits all that close to home. But as you can see in this picture, and it's probably a little bit hard because it's smaller than what it ought to be, but there's tractors here that are blocking the street. So this is, while I was in Obregon, there was a problem with water, and basically it was a matter of farmers fighting for the water that they're used to irrigate their crops. And so in developing countries and in developing agricultural communities, these are problems and issues. There are major problems and issues that are faced daily, and it's a matter of feeding people, even coming down to feeding their own families. And so like this was called No Al Novillo. Al Novillo is a reservoir. The people of the Yaqui Valley, obviously it's a desert, it's in the Sonoran Desert. They wouldn't have agriculture if it weren't for water. And so the government is proposing to carry the water away from the Yaqui Valley to the city of Hermosillo for farmers there. And this was an embargo that the farmers placed. They parked all their equipment on the roads leading in and out of the city, and they wouldn't let anyone in or out. So they stopped all the trade that was moving up and down through the area until the government responded. So just to let you know, you know, these are major issues that happen, you know, not here in College Station, but worldwide. There are major problems, water being one of them. So this Borlaug Fellows Program, there are two components to it. One is the Graduate Research Fellowship Program, and then the Summer Institute on Global Food Security. So the component that I participated in, and what I'll be talking to you today about, is the Graduate Research Fellowship Program. The other component, the Summer Institute on Global Food Security, is a 2-week-long learning program for graduate students attending U.S. institutions who are interested in developing a holistic understanding of agriculture on a global scale. And so it's a program that you apply to, and then once you're accepted, you know, they take care of your travel, they bring you up, you're invited to speakers, you're invited to talks, to listen to world leaders and speakers on the topic of food security. With the Graduate Research Fellowship Grant, the grant is focused on Global Hunger and Food Security Initiative, the Feed the Future Initiative, and it's, when you say food security, it could represent anything from agriculture, nutrition and ecological resources to poverty. So it reaches very far. It has a very broad context, which allows the grant to be opened up to a lot of different opportunities and focuses. So the objectives of it, as outlined by Purdue, to help train a new generation of scientists fluent in the problems of the world, basically, and to strengthen the capacity of the developing countries, to support key research themes based on the Feed the Future initiative, and to increase these understandings and links and to create collaborations. It's designed to foster cross-cultural collaboration and dialogue and produce long-term relationships that can move on as we go from being a graduate student into a faculty member at some point in our communities, or in our careers. And to work with international partners to sustain global growing and learning communities. So I'm not going to read all this to you guys. The print's a little bit small, but I just wanted to give you kind of an idea of what the focus was with the graduate research fellowship. So with this U.S. Boer Log Fellows in Global Food Security, it's designed for developing a component of the your graduate research project in a developing country, and typically in collaboration with one of the international agricultural research centers or the National Agricultural Research System. So one of the focuses was international research collaborations between U.S. scientists and then scientists at these centers, and then the awards are made on a competitive basis. So it's a grant. It's a full grant that you have to write. There's an RFA. I've printed off a few copies of it and left it laying on the table for you guys to look at. There's an RFA that's out right now that's available for it. But it's funding that you as a student write for. You compete. You submit a full proposal just like the Monsanto Beach and Boer Log, and then it's reviewed. And if the proposal's funded, then the funds come back to you at the university, and then you can use them as that component of your research project. So the grants range in value based more or less on the amount of time that you're planning to spend in the country. So for a 6-month stay, it's $15,000 US, a year, $20,000, and for a 2-year-long international project, it's a $40,000 grant. And so they require that you to be in the country for 85% of the time during those time periods. So like in my case, my grant was 6 months. I think I was there something like, I don't know, just short of 2 weeks of that 6-month time period, but because it fell within that 85% time period, or 85% block, it worked just fine. So the grant is not designed, it's a travel grant. It's not designed to cover the cost of your project. There is room within it that you can use for different consumable materials, shipping, all those kind of things outside of your room, board, and travel costs. But it's not designed, the idea of it is that it's partnered with a project that you already have in place that's funded as far as a research objective or an idea. One of the changes that they've made to it that wasn't part of it when I wrote it, or I would have written mine a little differently, was that students that are applying for year-long research grants can split it up into 2 6-month travel venues. So for those of us that are breeding students, it works out really well. So instead of only being able to go look at your population in a trial on the other side of the world one time, it allows you to go for 2 full seasons, but only have to apply for funding for 1 full year. So, like I said, the RFA is currently available, and it's open right now. The application deadline is Monday, November 11th. It's a 2-page long proposal, $15,000 for 6 months, anywhere in the world. Just going to throw that out there. So some of the locations that you could consider, that you could think about for this, or any of the CGIR centers. The one that I worked at was here at CIMMYT in Mexico, so it's really close to home for us. There's 15 of them across the world, and any of them work. The Boer Log Fellows website lists 20 participating partners, which are outside of those 15, which means that they're accepting applications or letting you partner with just about any program in the world to be able to work towards global food security, and again with the National Agricultural Research System. So now that you guys have kind of a general overview of what the grant itself is, I'm going to kind of switch gears a little bit and talk about the project that I applied for, and then the time that I spent in Mexico. So I was going to try and bring a slideshow of pictures of all the good times and everything else, but I thought it might be more effective if anybody's considering writing for one of these, that they'd be able to see how I put mine together, so that way it could kind of help you to formulate your own. So with CIMMYT, CIMMYT is the International Maize and Wheat Improvement Center in Mexico. So their mandate is obviously corn and wheat. And so this is also the center at which Norman Boer Log did his work. And so to alleviate a little bit of confusion, because this kind of gets hairy unless people are well aware of it, this is Texas A&M. This is CIMMYT El Baton, which is their headquarters, which is located just outside of Mexico City. And this is CENEB, which is Camp Experimental Norman E. Boer Log. And so that is in the Yaqui Valley in the Sonoran Desert. So it's a little bit further north, but it's a completely different environment than what we're seeing here. So in bore logs, the shuttle program could move lines between the two to create different seasons. For us right now, or for CIMMYT today, the breeding program, the majority of the work is solely done in Ciudad Obregón. So on the project that I worked on, my research component was here in Obregón, and then on the tail end of my project, I spent a month in Mexico City working with their biostatistics unit to try and work through some of the data that I collected while I was in Obregón. So if you wonder why Norman Borlaug chose that location, I give you this. And I realize that it probably has a lot more to do with the fact that the Yaqui Valley, at the time when Borlaug started there, but it wasn't, but now, is the highest producing location for wheat yield in the world. So it's a lot of sunlight, it's a lot of great temperatures and soil conditions, but like I said, there is no farming without irrigation. Looking at this picture, it reminds me of a talk I listened to a couple years ago by a wheat breeder in North Carolina. His name is Paul Murphy, and as a breeder, springtime is like the time of year to be in the field. And he made the comment that spring was his favorite time of year because it was like opening 1,000 little Christmas presents. And for anybody that spent time doing any crosses, that's kind of the same sentiment that we all carry. One of the big factors that makes the CENAB a great location to be able to do research, well, is this. It's based on the fact that you have a great environment, you have lots of water to be able to grow these plants, but because of that, it allows you to create different environments within that same environment. So here, this is a yield potential trial, meaning that the trial is giving everything it's wanted. The CIMMYT's very well staffed with very capable people. I mean, clear to the point that, like, the trials, 3 o'clock in the morning, they would go out and do pressure bombs to check the water potential in our lines or in our trials to make sure that plants were A, not under drought stress or feeling a drought stress, and B, not having too much water within the field. And so the trials are very well maintained. They're very well taken care of. So you have a yield potential environment. Here you can see by changing the planting date, we can introduce a heat stress. So by sowing a little bit later, to the same population one a little bit later than the other, as the plants come on and flower, we have a heat stress factor that we can put into it. And then also, of course, you just remove the water and then you wind up with drought. And they get the same kind of cracking in their clay soils that we get here when they're in drought scenarios, clear to the point where I think you could probably lose a cell phone if you dropped it in there. I think the most important part about the station and about the center is probably this picture. And this picture it was titled well by Dr. Reynolds, Matthew Reynolds, the PI that I worked for there. He called this bottom-up research. And so what you're looking at is one day we all had to go out and we planted 56,000 entries in one field by hand. And so I think the important part to realize about this is that it's not just about people and having large amounts of people, it's about having large amounts of very skilled people. And that's, I think, the biggest strength that CIMMYT has as far as their facilities and as far as their breeding programs go, is that there are a lot of people that understand how the systems operate, and it helps a lot when you're there trying to collect data to not have to focus on the day-to-day agronomics of your trials. So the program that I worked in was in the global wheat program, specifically wheat physiology. It's split there between wheat physiology and the breeding program. Kind of similar like what we have here with Dr. Hayes and Dr. Abraham, with a lot of us in the program. So in the wheat physiology, their focus is to try and adapt tools or adapt technologies to basically aid the breeder or to speed the breeding process. And so here you can see some pictures of some of the stuff that we were working with, just kind of a quick overview. Thermal imaging. This is a ASD. Mariano's wearing what we call an ASD spectral radiometer. So he's taking whole plot spectral radiograms right there in the field. And it looks kind of crazy when someone's walking through the field with a CPU on their back and a laptop hung around their neck, and they're taking data. But it's very high, very usable, reliable data. And I think even part of Fatima's project uses some of this technology. This is Jared. He's a Monsanto Beach and Borlaug Fellow down there at CIMMYT. He's working with Jesse Poland, and this is what they call the phenocorn. And it's it's an NDVI green seeker that they cruise through the field, and they're using corrected GIS data as they move through the field, and then also using an IRT to take canopy temperature. And so it's all data that they're taking over time in these multiple environments. So like in my trials, I have one population out. That means I have 3 fields that I'm taking data on every single day, because you have multiple environments within the one. And then this was kind of a cool project that Simmet's jumping into right now, is they're working with in crop modeling. And so they're working towards these ideas of forward modeling. They just hired a crop modeler, Phil, and this project was a lot of fun. They have these things, these audit crops, where they take canopy temperature constantly. They feed the information back to these panels. These panels make a cell phone call, and then they upload the data back to Simmet. And so we get things like light interception, canopy temperature, those kind of things. And they're all tools that are used for modeling purposes, as far as testing models, and then also in forward modeling scenarios. One of the funnest parts of the projects that I think that they have in physiology down there is the UAV platform. So they have an entire unmanned vehicle system or platform that they work with. It's headed up by Maria Tataris. She's this is Maria here. And so the 2 main components of that, one being the blimp and then the other one being the remote controlled octocopter. So they have multiple cameras, they have multiple tools that they go out with. They can take NDVI, thermal imaging. This camera that's shooting on the blimp is it's like 12 cameras in one. It's the same kind of thing, same kind of data that we receive from a satellite. So they have a lot of tools that they're trying and they're testing to see what kind of data and how far and what they can do with that. So all that said, I'll tell you guys about my project, or my proposal, really quick. The physiology group, I opted to partner with Dr. Reynolds because our lab has done work with him in the past on beach and boreal fellowships, but also he's very open to new ideas, and sometimes it takes people who are very willing to let you chase down rabbit holes to be able to make some of these things happen. And so the project that I applied for and that got awarded was titled Estimating Wheat Root Biomass Using Ground Penetrating Radar. And before I get any further with it, I do want to say that I'm not going to get into the data. I'm going to just kind of breeze through the topic and how the system works and what we're doing with it. But on October 9th, I'll be giving my seminar in soil and crop sciences. I can promise you it'll start with a laser show. I'll ride in on a unicorn, and there'll probably be fireworks at the end. So you should come and listen to the data and have a look at it when I go through it. I'll also be presenting the same thing at the ASA meeting in Tampa if anybody's interested to come to it as well. So the hypothesis of the project was that we want to use remote sensing technologies, specifically this one of being ground penetrating radar, to allow breeders to phenotype and select optimal root biomass and structure in breeding populations. So the objectives of the grant that I wrote was very simple, was to determine the correlation of root biomass using ground penetrating radar with direct field measurement techniques. So to give you an idea, a general idea of what ground penetrating radar does and how it works, because it's not real common to most, is I like to use this picture. This is a picture of well, let's go back to World War II. Ground penetrating radar works a lot like regular radar works. You send a signal, it bounces off something, it comes back, and you receive it. And they time the amount of time it takes for it to go and come back, and that tells us what that distance is. And then it gets a lot more complicated when you want to try and figure out what it looks like and those kind of things. So in World War II, the technology was really that simple. The planes were flying, and they would bounce the signal off the ground, and it would come back, and that's how they would tell what their altitude was. Well, then all of a sudden, we had all these planes that were flying over top the polar ice caps, and they were running into the glaciers. And they couldn't understand why this was a problem. And so this is based on where the idea of ground penetrating radar came from. What was happening was these planes were bouncing their signal, and they thought they were hitting this top piece, which was the ice. And then they were calculating their altitude, and their altitude was off, and they were running into these glaciers and inside these glaciers. But in fact, what was happening was this signal was not bouncing off the top of the ice. It was penetrating down to the seawater or even to the seabed, and then coming back. So at the polar ice cap, this could be a mile thick. And so they realized they could penetrate the ice. And what it was, the same principle that allowed them to do that and allowed them to understand that was the same principle that we use to be able to image things with ground penetrating radar now. And that's relative dielectric permeativity. So it's not, before I go any further, it's not a matter of how dense something is. It's how its relative dielectric permeativity reacts. And so what an RDP is it's how well something conducts a signal. And without getting into a lot of the detail, what I can tell you is that there are differences in RDP based upon the way what things are made of. So air has an RDP of 1, ice has an RDP of 3 or 4, seawater has an RDP of 80, and then saturated sands 20 or 30. So as you can see, it's not a very refined system. So differentiating RDP of 1 and 3 and 4 versus 80 is really hard to see. But you can definitely see the difference between 80 and you can see the difference in 1. So what I'm getting at is that same concept is what allows us to use ground penetrating radar to image roots. So before I go any further with that, I have to go and tell you exactly how horrible it is to work in roots in wheat or in any other crop system. So traditionally how this happens, or what happens, is the plant grows and we come in with a tractor and we take this soil core and it drills out a hole that's about 2 inches big, and we go all the way down and we bring it back and we section it into different sections. And then we take these samples and we flush them into this machine. This is basically, we call it an octopus, but it's a system that has air and water and jets that wash the mud out, and then it blows air into the bottom and it moves those roots up to the top, and they're screened off in the top. And so this is how we measure roots. Then we go through and we pick them all out by hand. We put them in a refrigerator, we do our analysis, or we dry them and we weigh them. So it's very time consuming. Obviously something that we're not going to apply to a big panel, especially in a mapping population or in a breeding program. Ground penetrating radar, back to the idea of relative dielectric permeativity. So as we bounce our signal down into the ground, you can see that we have these roots here. So the roots we're assuming that the roots have water in them, especially in a drought-stressed situation or scenario. More water than what's in the surrounding soil. So the RDP of surrounding soil is like 15. The RDP of the water that's within the roots is like 80. So that's what allows us to differentiate and see those interfaces of where those roots actually lay. So when we're looking at this in the ground penetrating radar, we get back this radiogram, and it's not a picture of the architecture or the structure of the roots. What we look at are the total masses of roots that are under these plots, and then we're able to compare those masses one to another. And so we can tell things like rooting depth, we can tell how large the area is, how far it spreads out from the plot. If we planted one wheat plant, we'd never see it. But in our replicated trials and in our field conditions, we have multiple, thousands of plants within one plot, and so we assume that they're genetically the same, that we have that representation is what we can use for their comparison. So as this antenna moves across the field, we ping a signal down. The signal goes down, it bounces off of something, and it comes back. So it's not one signal every few seconds. This thing fires in nanoseconds. So we get millions and millions and millions of returns that come back to us on the antenna as we're moving through the field, and it happens very quickly, and it's very high throughput. So what happens, like in our scenario in Mexico, we have the plants are planted on these beds that you can see here. And so we can take our root course for comparison, but what we do is we pass with the antenna here and we pass with the antenna here, and we determine where the outside edge of this is as compared to the outside edge of this. And then we use software that models the data between this point and this point to create this platform between the two. And so what that allows us to do is take the outside edge of both, and then we let the software make the assumptions between them as to what's connected and what areas is working within the two. What we get back after we model these platforms is something that looks like this. This is a three-dimensional point cloud of the data, and so this is an entire field, this is an entire trial, it's not just one plot. And you can see that you get these masses that are represented. This is some of the older data that we took, not this past season, but the season before that. And it's hard not seeing the actual plot measurements laid on here, but to be able to determine which are which. But we can also go into, let me go back here. We can also go into what we call sections or time slices of these platforms as well. So here, this is the soil. This is a few nanoseconds down. So nanoseconds in time equates to depth. They're both the same thing. And then we separate that out. So as we move through, it's kind of like a CT scan where as you pass through it, you see a little bit more of it and a little bit less of it as you come back through. And so we can take those and we can make, each one of these is a picture of the entire field. So each one of these, this is, a few nanoseconds, and then, 5 nanoseconds more, 5 nanoseconds more. So it's kind of as we're moving down through. So when we analyze these, we analyze the amount of root area that's in each one of those in each one of these planes as we move down through it to increase our statistical power within the data set.
Sean, I'm going to interrupt you.
Yeah.
If anybody needs to be somewhere at 1, if y'all want to go ahead and leave right now.
I'm almost done. There's only a couple more slides. So the project, the data that we collected in Obregon was on multiple populations that CIMMYT has put together. Recently there were some papers that were published that showed that contrasting canopy temperatures, the plant's ability to cool its canopy correlated to the amount of root biomass. So we built panels we put together panels based upon that information, and those were the panels that we used in Mexico for this population or for the data that we're working on. So that was my project. And I think one of the things that I want to say, and I guess the last couple slides here, is that they're the Borlaug Fellowship is very interested in the science, but they're also equally as interested in the broader impact at which your proposal comes in on. And so I think one of the things that sold my proposal was not just the fact that I had some idea that I wanted to push forward or a project that I wanted to do. It was that I incorporated multiple other components while I was in Mexico, and I think that helped to push forward my proposal. One of those was the International Wheat Yield Consortium. It was held in Obregon. So I attended and participated in it. The Wheat Yield Consortium is a network of worldwide scientists that come together, and their goal is to achieve 50% increase in genetic yield potential of wheat within the next 20 to 25 years. And so it's a platform of over 100 scientists that get together and they discuss their frontline technology and their frontline findings with the hopes or the idea of being able to move forward wheat yield and not just specifically the sciences or their CVs. This is the group photo that was taken in Obregon. This photo, I stuck it in here because I like it a lot better. This is the photo of the group, but it was taken from the octocopter as we were all standing in the field. So it was kind of cool that they were able to shoot a picture with that. My participation in that was I presented a poster at it, and then I also gave a field demonstration on the technology that I was working with. The posters in Mexico were a little different than what we're used to here in the United States. So it seemed to me more like a flyer, but that was kind of the format. So I don't know, it's just a little bit different as to how they do it. But it's it's a learning curve, and it's all part of the experience, I guess, is in doing things that are a little bit different. Another component that I participated in was the Visitors Week. Every year CIMMYT holds their Visitors Week. This year they had 157 visitors from 37 countries. If you look real close, you guys might recognize this guy. Dr. Ibrahim came down to visit me while I was there, and I think he attends this on a regular basis. So it's a chance for CIMMYT to showcase their varieties and their research, and it's also an opportunity for leading breeders within the field in the world to come together to discuss format and to discuss future directions. Another option, and then another opportunity that I had while I was there was I also got to participate in the modeling response to high temperature workshop. This was held in El Baton after I left Obregon and made the trip down to El Baton. I added this down here because this was the headline that CIMMYT had on their blog, was International Scientists Compare Hot Models at CIMMYT. So this was really a neat meeting because standing in that room, I was way out of place. Every one of these people that were standing there had authored or developed or built a published crop model. And so to be able to stand there and meet all the people, and what it was they all came together, they took one data set, they ran that same data set against all of their models, and then they compared them. And then the purpose of the discussion was for them to understand where they diverged, why they diverged, and how each model was different, and what was working and what wasn't working. It was really an excellent opportunity, but for me it was awesome because I just was a mouse on the wall. But it's one of those things that being in the right place at the right time, you get to really meet and interact with some of the neatest people that you could want to as far as in your career. So that was another opportunity for me. a couple of things for that. This Borlaug Fellows Security, or U.S. Borlaug Fellows Security and Global Food Security, I have a poster that will be at ASA if you guys want to check it out. And then I had a publication with CIMMYT that came out in 2013. It was basically just an update on the opportunity in the project, very similar to the talk that I'm giving to you right now. So with that, I can take questions, and before I do, I might add, if you guys, if I blew through something or you want more information or there's something that you want from this presentation, it'll be available on my website, precisionphenotyping.com, and you can go there and download it and pretty much any other talk that I've ever given in the past. So with that, please apply. If you have any questions, please ask me, or, and if, I'm even willing to share with you the proposal that I wrote, because I think that this is an opportunity that creates lifetime friendships, especially within our careers and in our breeding programs and such. Thank you. Any questions? Yes.
Sean, you'll cover it in more depth, I'm sure, next month. Can you touch on the efficacy of this system based on soil type? And also depth, those of us who work on crops with deeper roots.
Yeah, so he's asking about the GPR project. So it has a lot of limitations. This is no out-of-the-box system whatsoever. One of the biggest limitations and what has stopped agriculture from using this technology and anything all the way up to this point is if you read the literature, it's all going to be unanimous in the same thing, that you can't use it in clay-type soils. And the reason for that is clay soils have a high capacity for water. That water conducts the signal. Remember our discussion about the RDP. And what happens is you fire the signal into the ground and it just keeps going, and it never comes back. And so you lose a lot of that information. And so you're unable to see the differentiation or the location of these different things. And that's one of the few things, or one of the few problems that I would say that lie with using ground penetrating radar, that we've changed and made some adaptations with it that allow us to visualize. One of the problems I had with this project when I first started was I wanted to change the way we grew wheat, not the way that we use ground penetrating radar. And you can't do that. You have to do it the way that farmers do it, and then adapt the tool to be able to look at that. And so that's been kind of a challenge. With regards to depth, the depth of the signal is based upon, and it's a general guideline, but it changes with every soil type, is the further you go, the less resolution you get within the first few feet. So for us, we're using a 1.6 gigahertz antenna. We lose the first probably 3 centimeters of resolution, but it only gets down to about 40 centimeters. Versus a 400 megahertz antenna, it'll get down to, 10 feet, but you lose the first 10 inches. So, it's different based upon the soils that you're in. And one thing that you have to keep in mind with that is that can be different even within the same field. Everybody's seen like an EM map or seen the differences in their soils. Salts have a big effect on this. So spatial variation within the field, if you're not compensating for that or selecting your locations based upon it, it gets kind of hairy. And so as a high throughput tool, I don't know that ground penetrating radar this year or next year is going to be something that we're pushing through every one of our field trials or every one of our plots. But I think that it works well in the idea of being able to select, like with the previous talk, parent lines. Being able to look for those that have real deep capacity and those that have shallow capacity, and looking for those for that contrast if that's the population we're trying to map or what have you. Any other questions? Awesome. Thank you.